we have an aquarium where you have a range of organisms in there, including pondweeds, newts, and some other animals. And two things are being measured in this aquarium. One is pH and one is light intensity. The first question is how long the experiment was conducted for. Determine, well, that's not the first, it's the second question. Determine how many days the data logging covers. How would you get that answer? I know the book's answers are available. If you've looked it up, you could have it in front of you. But how would you know how many days the experiment was conducted for? Ananya, do you know?
No, sir. I guess the time in the graph.
Well, yes, but what does that, what axis are you looking at in the graph?
The x-axis.
X-axis, yes. Do you have any information there now that you can understand from what you're seeing in the x-axis to tell me how many days the experiment went on for? Tell me if you're not sure or you don't know.
I don't know.
Okay. Let's go to Alicia. Alicia, can you tell how long the experiment is going on for?
6 days.
6 days. How do you arrive at that?
On the end of the x-axis, you can see that it says, like down says absolute time, DHHMMSS. Over there it says 6 days.
Right. So exact. So tell me in terms of days, hours, minutes, Alicia, how long is this experiment running for?
6 days, 2 hours, 45 minutes, and 9 seconds.
Right. So Ananya, you are seeing now what's happening there. You notice this is the explanation. You get it now, right?
Yeah.
So days, 6 days, hours, 2 hours, minutes, 45 minutes, seconds, 9 seconds. So this is a data logging device that can measure time, and it's also measuring how pH level is changing inside of the aquarium, and it's also measuring the light intensity for us. Okay, so this was really designed to look at daytime and nighttime effects, really, but it's a whole controlled system, a little bit of a closed system. And this is, of course, what the mesocosm is all about. It doesn't have to be having a lid on it necessarily, but kept under controlled conditions. A small piece of our natural environment is brought into the lab and put under controlled conditions. The controlled conditions here is we have decided that we're going to take it through a cycle of light and dark every 24 hours to simulate day and night. Okay. Now, Dia, what would you know about plants if it's given light? What gas would the plant give off if it's given a lot of light?
Oxygen.
Give off oxygen. And what gas would it be consuming a lot of?
Carbon dioxide.
Carbon dioxide. So now, when there's a lot of light in the system, can you tell me a spot where there's a lot of light in the system? Okay. Don't know if you...
Any one of the spikes in the red.
Any one of the spikes in red. Good description. So like this one here. Now, there's something happening with the spike in green also when there's the spike in red.
Yeah, when there's a spike in red, the pH increases in the green place.
Right. Now, that is a very deep analysis that we are going to be doing here, because that spike could be misleading. That spike in green could lead you to think that something is really increasing. But something is not really increasing there. Of course, you guys may have the answers now, which you could have, right? Because I've shown you how to arrive at the document that carries the answers. But without going there, something has increased. What is that thing that's caused this increase in pH? That's the question that we should really reflect on. And we have to reflect on it with a bit of knowledge of chemistry. And even though many of you are not taking chemistry necessarily, some understanding of chemistry is a requirement really to to progress in biology. Whether you took chemistry before or not, you still have to actually read up on quite a bit, and knowing about the pH scale is one such thing. So if you don't know about the pH scale, it's something you should read about, because it's definitely essential in science and in biology that you understand about it. Okay, so Shreya, what do you have to say about why the pH has gone up when the light intensity is high?
So the reason why the pH goes up when the light intensity is high is because the rate of photosynthesis goes higher. And so when the rate of photosynthesis is higher, that means that lots of the CO2 that's inside of the aquarium is being used up. And when the CO2 is being used up, it means that the pH is going to change from being more acidic to being more basic.
Right. So that is the correct explanation, and it's recorded because it's very accurate. But what could be misleading is we might see that, you know, the green arrow is the green line is a peak, right? But really it symbolizes what? A fall, isn't it?
Yes.
Isn't it, right? It's a fall in CO2 that's being reflected by an increase in pH. That increase in pH, of course, is being caused because CO2 is being taken out of the aquarium. Now, what are we able to conclude about adding CO2? If CO2 gets added to water, what's going to happen to its pH? Not you, Shreya, let's take someone else. If carbon dioxide is added to water, what's going to happen to its pH?
The water becomes more acidic, so the pH decreases.
Right, the water becomes more acidic. So, Sahana, where do you see that happening in the graph? The water becoming more acidic after becoming basic? You see it happening here?
Just 1 second.
Can you guys see my mouse on the screen, by the way?
Yes, sir.
Yeah.
Right. So is it here that you say the pH is falling? It's becoming more acidic. Would you agree with me on that? This spot right here?
No, that means it's getting more acidic, sir.
Right, it's getting more acidic, but is the pH falling?
When the
Yeah.
falls,
Yes.
gets more acidic.
Yes.
Yes.
Yeah.
Well, the
Oh,
pH.
because it's going from 7.35 closer to 7.30. So
Right,
yeah, it's falling.
it's becoming more acidic, and that is when CO2 is actually it's not as low as it was before. It's actually increasing. Okay. Now CO2 is going to increase. Carbon dioxide is going to increase when what happens in the aquarium? So what has to be happening in the aquarium?
Photosynthesis decreases.
And what increases?
Rate
Respiration.
of respiration.
Rate of respiration. Now this whole activity is just reminding you of what you know about the carbon cycle. But what it's doing is it's taking the carbon cycle and it's bringing it to a connection of your understanding of the pH scale, and also a knowledge of when you pump carbon dioxide into water, the water becomes acidic because you are creating the weak acid, carbonic acid. So this experiment that we have here on the screen, it could be done at CIS. We could set up our aquarium with a light sensor and a pH sensor inside of the aquarium, or we could create our own small little mesocosm. You guys can create your own small little mesocosm at home, put in a lot of plant life in it. And if you want, or if you already are in the business of keeping aquariums, you might have a pH sensor. pH sensor is not an expensive thing to get, a simple one. And I'm not saying it because I want you to get one, because we have at CIS. If you want to get one as something that you want to have, then you can put it in there. Or you can just buy some pH paper, and every few hours you can test the pH with pH paper. And you can regulate your light intensity. You don't need newts or other animals or anything like that. You just need a lot of aquatic plants. And you regulate and you see if you can carry out this experiment. And you might want to control certain things in there, and then other things, and ask some bigger, slightly different questions. And suddenly you find that this is rigorous enough for you to write a report on and let it become your final independent investigation. Now, I don't want you guys to decide on what that investigation is yet, because we want to go through many topics. But the way that you arrive at what you're going to do for your IE is not by looking on Google to see what can I do for my IE. Unfortunately, that's what a lot of students do these days. And with this syllabus going on for about 5 years now, there's a lot of previous IEs already posted. But the way that it was meant when it was created by the IB was that students would, in reading and in doing activities, you would end up asking certain questions that you feel you're very interested in. And that's where the personal exploration would come in, because you were reading this or you were experiencing this or we did that activity in class. And now you want to investigate your own thing. But this fluctuation continues to go on and on, like that's the same pattern really, pretty much. The light goes on, the CO2 level falls. Make sure you're following that reasoning. This green line is not CO2. This green line happens because CO2 falls. And when CO2 falls, pH becomes more alkaline because carbon dioxide makes the water acidic. And when we take it out, the opposite of acidic would happen, which is to make the water more alkaline. And that is basically what's happening throughout in this cycling that's going on. Okay, and that's the answer to this question. Explain this trend, deduce the trend in pH in the darkness, and explain the trend. And what this is doing is it's just introducing a practical investigation applied to what is really a mesocosm, which is a system that might be open on the top, but it's an open system mesocosm, where you control a part of the natural environment and you make certain conclusions and you get experimental evidence to show that these aquatic plants are taking in carbon dioxide when they get a lot of bright illumination, and they are just respiring and surviving once the lights are off. Everything begins to drop and the cycle continues to go on and on like that. So an important question for you guys to take a note of, and of course we are taking a note of it here by recording it. Any questions about this one? All right. So I'm going to stop sharing this one now. I just want to see that everybody has rejoined, because Kartikeya, you were having some problems, it seems. Are you hearing me out there now? Not hearing me? And what about Sanjana?
So I'm here.
Okay. All right. So let's move to another question now, guys. And that question is going to be, let me stop presenting here. I'm going to present another window now. This window has an experiment. And the very important activity is this one now, especially because we are not at CIS able to set it up. Again, the mesocosm is the center of things. And this is where I want to stress that the mesocosm is not just that first activity. That's not, if you are in a school and the teacher tells you the mesocosm is just you put some soil and you put some plants in there and then you get that out of the way and we don't talk about mesocosms again, it means they're kind of misunderstanding what this mesocosm thing is about. Because kids in 3rd grade can get that assignment, what you guys got to start off with, right? And they do get it. And they enjoy it a lot, and they could talk about the plant needing light and everything, and the water cycle. And you guys were asked to talk about the carbon cycle and everything. But the concept of the mesocosm is really for the environmental biologist or the environmental scientist to take a small piece of the natural environment again, like you have in this bottle. Seal it in this case, so you get a closed system, and bring it under controlled conditions. In this case, just like in that last experiment with the aquarium, but this one is not an aquarium. This is a terrestrial plant, a land plant. And when we're dealing with a land plant, we can't measure CO2 with a pH sensor. So we need something to stick into the air. And can you guys see where my mouse is right now on the screen? Let me hear.
No, sir.
Okay, you can't see it on the screen now. Let me go on the other screen and see if you can see it on that side then. Now can you see it? Can you guys see my mouse now?
No, sir.
No, sir.
Okay, so for this sharing of this window, it's not happening. But there's a big rectangular box there at the top, right? A black little box that's sticking into the bottle. Can you see that?
Yes, sir.
Right, that's your CO2 sensor. And then, of course, there's the light sensor. Both of those sensors are up at the top, showing you something similar, very similar to the last experiment. In fact, it's pretty much identical to that last experiment that we looked at. Just now, this is a terrestrial system, and that was an aquatic system. So now we are going to generate some data for this experiment. We're going to speed it up. This experiment, again, just like that one, happened over 6 days. This one is happening over several hours. And I'm going to start it, and you're going to see data appearing in all 3 sections. But before the data appears, if we are going to change light, and you'll see the lights will get switched off, we'll put the cardboard over by the window, lights will get switched on and off just like the aquarium. When the light switches off, Dia, no, not Dia, Wuta, when the light switches off, Wuta, you hearing me?
Yes, sir.
When the light switches off in this, what do you predict would happen to CO2 levels?
They would increase.
Why they would increase? It's
because then photosynthesis, the rate of photosynthesis is smaller than respiration.
Yes. So what do you predict would happen to oxygen at that point, Wuta?
That would fall.
That would fall, yes. So you have made a prediction. You have made that prediction because you have understood some science before.
Yeah.
And from the previous activity that we looked at and from before that. And now, when somebody sets this system up, you can predict what's going to happen. But predicting what's going to happen is one part of science. The other part is, of course, when you say that to someone, Wuta, and this is related to TOK, guys. When you say to someone, go to your church or your temple or your mosque and pray every day because you want COVID to go away, and if you pray, COVID will go away. You just have to take that with faith, because nobody can prove to you that that prayer is ever going to help. Okay, but that is a way of knowing that people have come to accept that as a way of knowing that you can pray for something and you can have faith. But the difference between that, Wuta, and science, of course, is you have made a prediction. You have told me something, and I think you're correct. But I'm not saying that you're correct just because I'm biased to you, or I like you, or we believe in the same religion or whatever. It's not like I'm saying it because of that. I can set up an experiment. I can set it up in an aquarium. I can set it up in a huge terrarium or a small terrarium like this one. And then we can get results like what you're going to see on your screen now. So take a look and let's see the experiment begin to happen, you guys. So it's happening on your screen there in real time. Look at what Wuta predicted. Light is falling. Light is falling there. And now we see what's going to happen with CO2. Light's dropped to nothing, and suddenly, then you start seeing the spike. In the green, the carbon dioxide, and the blue, the oxygen. You see that inverse relationship between both of them. As one goes up, the other one falls. And you guys, from what I've been hearing from our previous discussion and from what Wuta just said, everyone who's been participating, you understand why this is happening. You can explain why this is happening. You can set this up when you come to CIS. You can set up something simple like this at your home with aquatic plants. And you can show that this is happening. And this is what the nature of science is. We can understand it. We can make predictions, and then we can carry out experiments to verify those predictions that we make. So the scientific way of knowing is very much based on evidence, hard evidence that we can present like this. Sometimes, of course, there's evidence that the evidence is not as hard and as easy to generate as this mesocosm, as we are going to see in topic 5 when we deal with the topic of evolution, which is an area of science that where the evidence is not so easy to just say, I'm going to show you how life on Earth arose over 4 billion years. And in that case, science needs a little bit more indirect kind of evidence. For this one, you have all the evidence you need to understand the cycling of carbon dioxide and oxygen in ecosystems. Right? So, any questions about it? What we have is this recording. What I want you guys now to do is, when this recording is posted, to take the graphs. If you're not sure about what's happening, write annotations on them to make sure you explain what's happening, because this one, it's really identical to the one from the book in terms of the concept that's being presented, but it's being presented with 2 different mesocosms, one being aquatic and the other one being terrestrial. And we'll end up with one last discussion today. And I want to again check and see if everybody is here, because I'm concerned that Kartikeya has lost his way out there in the internet. Somewhere he's having problems. So he's not there. So I'll have to contact him.
He's trying to join, but he can't join because his internet isn't working. He just texted us.
Oh, he texted you, Ananya?
He texted on the Hangout as well.
Oh, he texted on the Hangout.
Yeah.
Okay. Well, once you guys are trying to join and whatnot, I know everybody's being honest about trying to participate. That's all we can expect. Fortunately, today we are recording this for the benefit of this group and for the other 2 groups. So he could come back and follow. Okay. One more window to share. Let's see. Suyun, I'm going to ask you to talk about this one. I haven't asked you to engage in the discussion as yet. So get ready. All right, Suyun, can you see the screen?
Yeah.
All right. Tell me what you understand. I'm not going to explain. What do you understand from what you are seeing on your screen right now, in terms of the topic that we are on and the heading you're seeing and everything?
Maybe you can say the input of carbon dioxide.
Okay. Yeah. No. Like when I ask you what you understand in terms of like what is all of this looking like to you? What's inside of the jar? What are the 2 sensors? The big pink block at the top. I want to know that you understand what you're seeing then. What's inside of the big container?
I can see a plant. I think, is that a plant?
It is a plant, right?
Yeah. I can see a plant.
What?
And I can.
Right. So what is the big pink stuff at the top of the page, the top of the image? Okay.
It's light.
It's light. It's a lamp, right? Now, what's the green thing that's sticking into the container?
I think it's input of oxygen.
Well, it measures oxygen. It's what we would call an oxygen sensor. Okay.
Yeah.
Now, this video image was not made at CIS, but it was made by me. And it's at CIS, we also have oxygen sensor. We also have a container that's specially made with a rubber fitting to hold that sensor in an airtight way so the experiment becomes valid and it's not affected by anything. Okay, so that it's this little special fitting. And then the other sensor, Suyun, this one here, what is it?
It's carbon dioxide sensor.
Right. So then you can see where we are going. We should be expecting something very similar to what we just saw in the other 2 experiments. But again, now I'm showing you guys another way of doing the experiment. This particular one is different from the one I just played because I am not going to measure what? What is it that I'm not measuring here? Who did not get a question? Alicia, I'm not measuring something. I'm measuring CO2 and oxygen, but the other variable that we have here that I'm not measuring it. Which is it? Compared to that last experiment, Alicia, there were 3 graphs, right? You remember?
Yeah.
What were the 3 graphs about?
One was light intensity.
Right. So is there anything here measuring light intensity as far as you see?
No.
You're seeing, Alicia?
Yeah.
Right. Nothing is measuring light intensity, right?
Yeah.
Okay. But it looks for all purposes like it's just like a rerun of what we had before. But it's got a little bit of a difference. We are going to take a little bit of a preview of topic 2.9 for IB biology as well. And I'm going to let someone have the joy of telling me why we have a red arrow, a blue arrow, and a green arrow. If you know, it's open to anyone. Why we have that? Okay. Dia, you know why?
No, sir, I'm not sure.
Okay, what about Sahana? Sahana, you have any idea?
No, sir.
Okay, now I'm going to give a hint then. It's about what colors plants of light plants prefer for photosynthesis. Did you guys ever recall reading that or hearing about it at any time? So
is it like chlorophyll can absorb light better?
well, certain colors, plants, most of the green plants that we have in the whole planet tend to use a lot of blue light and red light. Because you know what's coming from the lamp, it's just like white light, right? What's coming from the lamp is white light, and what comes from the sun is what you call white light. It's made up of all of the colors of the visible spectrum. You guys might have heard about red, orange, yellow, green, blue, indigo, and violet, Roy G. Biv, or Vibgyor, the other way around, as it tends to be described in India especially, but Roy G. Biv and other places. Red, orange, yellow, green, blue, indigo, and violet. And plants tend to use a lot of blue light and red light. That's what they want. But they reflect green light, which comes to our eyes, and then we see the plant looking as green. So everything around us looks so green because plants really don't want green light. They take the colors they want, they absorb that. That's used in photosynthesis. That's going to begin in 2.9 for IB biology, and it's going to connect to topic 8 later on. And again, you see what I had told, what the discussion I had about with TOK with Wuta earlier? That applies here again. You're not really doing any science, you guys. If I lecture to you and I give you this as a fact, and you write it down in your notebook, the question that you should always be prepared to ask me is, how do I know that this is so? And can I prove it to you? And if I can't set it up in the lab at CIS and prove it, which I can do for this one, I would show you at least a video of somebody doing an experiment somewhere else or writing a research paper in a journal somewhere else that proves it. Otherwise, it's not really science, because I can't make a claim and not be able to prove it with evidence. Then it's not science. So where is the evidence to prove what I just said? Here it is. And this, of course, is another practical aspect of biology, and it's important that we engage with it at least on the screen like this, because we can't go to CIS right now and do it. Wuta, are you seeing my laptop on the top there of this image? Where are you, Wuta?
Yes, sir. I can see it.
What do I have in front of the laptop there? What does it look like to you?
Like a device that can read, or like the light. I don't know what kind of device it is.
Well, it's a good assumption, Wuta. You see that it has like a little spectrum drawn on it here if you look carefully, right? A sample of chlorophyll is put in there in a little chamber called a cuvette, a plastic container. We do have one of these at CIS. This is a relatively expensive piece of equipment for high school science. It costs about $700 US for a vernier spectrophotometer. And you put it in here and you run this reading off. And this reading, does it prove what I just said, Suyun? Do you think it proves what I just said?
Yes.
Okay. Well, if it does, can you make a suggestion as to how it proves? This axis here, Suyun, I will help you a bit. It says absorbance. So can you tell me two colors that are really being absorbed a lot?
Blue and red.
Yes. And then what color is not really taken up too much?
Yellow.
Yellow, and then in the green area. And then it so happens, so if you look at the beaker actually looks the same color like this area, doesn't it? That area, and that's why it looks like that, because the things that are in that beaker, they don't want that color of light. They reflect it to your eyes and you see that. But the colors that they want to use, and when I say they, I mean all of the algae really, it's a lot of photosynthetic algae that I have in that beaker. They've absorbed the red and the blue out of the white light, leaving us to see the green. This is called the absorption spectrum of chlorophyll. Okay, so I'm just giving a little preview because it's related, and I've got it on this video for topic 2.9 for IB biology. All right, and then of course we come to this data. If this is the data that I have, Shreya, how can I say that my plant is getting light? What do I have to prove to say that my plants are getting a lot of light or not getting a lot of light? Whichever.
I guess when the oxygen level increases, as you can see in the graph on top, you can say that the oxygen level only increases when the carbon dioxide level decreases, and the oxygen level only increases when the rate of photosynthesis is higher than the rate of respiration. And so with that, you can infer that the plant is getting light to carry out photosynthesis and therefore let the oxygen level increase.
Right, good answer. But that red line that I drew there, are you convinced that that red line should be drawn like that? Look at the actual reading on the device. Any comments on that?
I personally don't think that's a line of best fit.
It's not a line of best fit, no.
Yeah, I don't think it is, because the graph itself is too all over the place. But if nothing else works, you could use that, I feel like.
Right. Well, what that does, it doesn't. And if you if I were out trying to prove this to you, right, right, Shreya, you would be within your rights to say, no, I don't believe you, because look at what happened there. Don't try to trick me. Okay, if you were a skeptic. And
yeah.
But you're not a skeptic. You know that there has to be some kind of reason, right? And sometimes when scientists go about their business like that, with some kind of bias beforehand, it is not really good for experimentation. When the scientist goes in, thinking, I'm just really here to verify what everybody knows. So it's really important in the hard practice of science that that anomaly there gets properly explained. And that's why it's so important that you, the scientist, understand your instruments that you're going to use. You know how to calibrate them. You know how to troubleshoot them and fix them. You know how to do what we would do at CIS. I would go and tell Mr. Roberts or Mr. Ranjan, they are our technicians, and they would call the people at Vernier and say, look, we need to get this oxygen sensor fixed or serviced, because that's a sign that it's not quite working properly. Okay? And it does tend to do that. It's a piece of equipment that kind of malfunctions quite a lot. And once you're aware of that, then of course you won't be under stress trying to explain the unexplainable, really. Not by the logic of the science that you are studying, but you are confounded by the technical aspects of instrumentation. And all of that is part of the practice of science. So it's not like sometimes students are set up to be frustrated in the lab. But when the frustration does come, it's part of the process as well, because no matter where you are and how expensive and fancy your lab is, it is a part of science that things don't always work the way it's predicted for a range of reasons. And you guys are seeing that right now with your little experiments at home as well, where we all had the same procedure, and for a range of reasons, people are getting different results. But we have one more question to discuss, and we are going to see this here. This question is for you, Suyun. Do you know the name of the, are you familiar with the thing that was inside of this box here? Suyun, are you still there?
Yeah, I'm here.
Yes, tell us about the thing that was inside of that box. Can you see the name?
Yeah, Pocky.
Are you familiar with it?
Is it something, is it, biscuits? No, not biscuits. It's,
So they're like sticks, I think, like, and they're
Yeah.
covered in chocolate.
Yes.
They're like
Suyun, I'm going to make it.
biscuit sticks.
Suyun, you've been growing up in India most of your life, correct?
No,
Where have you lived before?
Korea.
And
But
you never got Pocky over there?
No, I did.
Yes. So it's just a little side question there, Suyun. Because, you know, this experiment, I set it up myself, right? And I had some students from Korea who were running a club in my classroom. They used to come and use the room. And they had a sale of Pocky to raise some funds for their club. Okay? So that's why I picked on you to ask you that one. But yes, who is it? Sanjana?
Yes, sir.
Yes, Sanjana, you're correct. Do you get Pocky in India?
So you get it in some stores, but it's very rare. I think you get it in like Nature's Basket or something.
Okay, right. But it's pretty much a big favorite, I think, among the Korean students that I've taught, Suyun. But you are not so familiar, okay? But we shouldn't have a stereotype on everyone. That's not good, right? But yes, why am I putting a light on this box? And why did I put the box in the first place? So let's go back to Sahana. Why the box, Sahana? What are we looking to do now?
You're trying to like not allow the plant to get any light, so you're covering it with the box.
And you know you can predict what's going to happen with the sensor reading, isn't it?
Yeah.
You can do that. But why is the light left on now? Let's see. Let's open that up to everybody. Alicia, do you know why the light is left on?
No.
Okay. Bhuta, do you know why the light is left on?
No, sir.
Okay.
So is it like even if is it like to represent like the sun is there even if something's like some bush or something is covering up another plant, that there's still the source of light?
Well, that's a good try. That's a good try, Sahana. But I'm going to give you guys a word now. Control. So, and heat. Anyone getting the point now? A light bulb doesn't only give light. It also gives a certain amount of heat. So by leaving it on there, I am just allowing it to generate the heat that it generates. The plants aren't going to get any actual illumination, but whatever heat the bulb produces is still around. Of course, it's still being blocked to some extent by the cardboard, but the heat aspect is kept, because when you are doing an experiment, what I wanted to do in this experiment was just give light and give dark. I didn't want to change anything else. I didn't want to change the plant. I didn't want to change the temperature of the room. And if that bulb there was on and that was affecting the temperature, I wanted to leave it on, because when you are doing a science experiment, you change one thing at a time. That's your independent variable. And you keep everything else constant. And then you are measuring the outcome from that, your dependent variables. So, final question for the day before we are done today. What are we going to expect on the graphs? Are we going to expect the graphs to reverse themselves now, or look the same as before? Remember, what we had before was this. Let me go back. This is when the light was on. What should we expect now when the light is off?
The graphs would be the opposite.
Graphs would be the opposite. You are sure about that, Shreya?
Yes, sir.
Right? And you are sure about it once, and if it doesn't happen, you'll have to look for an explanation, because all of the other evidence and teaching and experiments that we've looked at today says that should be so, and I think you understand why. Let's see if you are correct. And there it is. And notice the oxygen sensor, Shreya, has behaved better this time, hasn't it?
Yes, sir. It's more uniform.
So both of them are lines of best fit. So it wasn't right really for me to be satisfied, but making these videos, you know, I have to set up so many things. A little bit frustrating when I didn't get that first graph how I wanted it, but that's part of it, and it allows me to discuss with you guys the truth about really what happens with these instruments sometimes. So given the fact that you guys are sitting at your homes, we can't go into the CIS lab and use all of this equipment which we actually happen to have at CIS. We did the next best thing today, which is to sit and discuss and look at the instruments and take it in and see how it's all set up and everything. But there's nothing to replace the reality of when you connect the sensor and then you come to me and say, sir, my computer isn't recognizing it, Wi-Fi isn't collecting, the Bluetooth isn't working. All of that is part of science. So science can't really be read from the book or discussed from a video, because there's so many unforeseen things that happen when the rubber meets the road, really. And it takes up so much time, really, in the science lab, that sometimes students feel it's better we don't do any labs. Teachers even might be feeling that, but then the true science teacher understands that the student must experience science. Okay, but right now we can't experience the full-fledged science, so we are taking on this experiment this way. But very important that you guys try to do your simple versions of scientific protocols at home, which is why Wuta, I told you I'm not worried if your seeds were frustrating you. That's part of the story as well. Get your seeds and get going. Have you arrived at any seeds yet, Wuta?
Yeah, they came just like an hour ago.
Okay, so take your time and maybe contact some of your classmates who have been successful. If you're not sure about what to do or you want to find out exactly what they did, that's also part of the nature of science, collaboration with other scientists, not to steal their work, but to replicate and to follow and to understand and to learn from it. So we have pretty much the end of our time today, guys. And next class, I want you guys to continue working on the other two database questions. I'll give some time. We'll start off with the Kahoot, and we'll get some guidance on how to write up the lab report. And I'll give some details and answer any questions about the unit test, which is next Friday. So one last call. Let me see if Mr. Pandey is around. Is he here? Otherwise, I will try to get in touch with him and make sure he gets access to the recording. Okay, guys. So that's it for today. It's lunch.
Thank you, sir.
All right.
Thank you, sir.
Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Bye. Bye.